Assalamualaikum and hi to everyone. Okay, first of all, I would like to congratulate to you because you almost finished biotechnology course and going to the LI latihan industry next sem. Okay, it's almost finished the course and successfully, inshallah. So for this reason, we want to do this session, which is a lab skill basic um, program to make you to remind you back or recall back what you have learned, the basic skills during the your course throughout your course. Okay, my name is SUHANA binti ADZAHAR. I was trained as a molecular biologist. Okay, starting from my degree, I'm doing biotechnology, and then a master degree and also PhD degree in molecular biology. So my work is mostly on the analysis of DNA as well as the proteins. Okay, the title given to me for this lecture is this slot is primer design and application of PCR for analysis of DNA molecule. Okay, this topic we have discussed it or touched it in the cell and molecular biology course as well as gene technology course and lab. So this is just a revision for you to recall back what we have learned. So what is PCR? PCR is a polymerase chain reaction. It's a technique to make many copies of specific DNA region in vitro from a single copy or very less copy of DNA. Okay, the purpose of doing the PCR is to propagate or make more of the template in the sample. PCR relies on a thermostable DNA polymerase, which is tag polymerase, okay, and require DNA primers designed specifically for the DNA region of interest. Okay, you can design primer whether it's homologous to your sequence or heterologous to your sequence. So other components needed in PCR, dNTPs is a building block of the PCR that you're going to copy, the DNA that you're going to copy. Buffers, okay, to optimize the reaction of the enzyme, water, and also DNA template that contains the target sequence that you wanted. In PCR, the reaction is repeatedly cycled through a series of temperature changes, okay, which allows many copies of the target region to be produced. Okay, from the diagram, you can see the step one is initial denaturation, okay, to get the correct temperature for the denaturation of the double-stranded DNA into single-stranded DNA. And then the step two, which comprises 30 to 35 cycle, okay, depending on the what the purpose of your research. If you just want to testing, you may stop at 30 times, or you want to optimize, you can go between more than that cycle. Okay, the step two comprises of denaturation at 94 degrees, and then followed by the anneling steps. Anneling is to allow the primer bind to DNA template. Okay, this about 50 to 65 degrees. And then followed by elongation, where the copies of the DNA template extended from the primer that you anneal to the DNA template. So step three is the final elongation period, where the unfinished copied DNA will be done here. Okay, so depending on the size of your expected PCR products, the longer the PCR product is, the longer the elongation it should be. Okay. Okay, primer. Primer is one of the components that needed in the PCR. Primer is also called oligonucleotides. They are short, single-stranded polymers of nucleic acid. Oligos may be unmodified or modified with a varieties of chemistries depending on their intended use. Okay, for example, here, if you want to have enable the ligation process or block the extension, you can add 5' or 3' phosphate group in the primer. Labeling with the radionucleotides or fluorophore and or quenches for use of probes, okay, for use to use it as a probe. The primer can be used as a probe as well. Okay, when it label, you can detect it during the process of the analysis. Okay, in the class also we have learned how to design a probes, okay, whether it's heterologous probe or homologous probe. Okay, make sure to check what is homologous and heterologous. Extension with other linkers and spaces of diverse functionalities. Okay, if you want to have an extra function of the gene that you express, you can link the primer, okay, the starting primer with the tag, okay, for easier end of process. Like you may put the his tag at the end of the primer so that you can detect your protein for the purification using the his tag. Uses of primers, okay, PCR assays, sequencing, DNA microarrays, okay, which involve the probes as well, antisense oligonucleotides, which will bind to the mRNA that produce in the RNAi technology. So this can silence the functionality of the DNA that you want to analyze. Okay, designing a primer. Okay, as you know, the oligonucleotides primers are necessarily when running a PCR reaction. One needs to design primer that are complementary to the template region of DNA. Firstly, in the designing of the primer, you should have the gene of interest sequence. Okay, gene of interest sequence, whether it's specifically, okay, you already have it in the gene bank, or you can also find the related or the same family or genus member of that particular organism that you want to study. They are synthesized chemically by joining nucleotides together. Okay, these services we can get outsourced from the other companies. You need to design the primers and then send the sequence to the company, and the company will produce the primer that synthesized, okay, by chemically to give you as a primer. The primer can also be designed by using bioinformatic tools such as primer tree, primer quest, primer selection. Okay, I believe we already discussed this in the GeneTech lab class, okay, where you use some bioinformatic tools, okay, to design a primer and also the DNA analysis of the transgenic. Okay, primers should generally has these following properties. Okay, length should be around 18 to 24 bases to ensure the specificity as well as the stability of the binding DNA to the template, the primer to the template. So the second, it should have 40 to 60% GC content. Okay, remember then when we do the primer design, we do the calculation on the how many G and C nucleotide in the primer. Okay, the number of total G and C divide by the total nucleotides in the primer, and then you can have the times 100%. So you can get 40 to 60%. Okay, if you first design the primer, you found that the GC content is much low, so you should add more G and C in your primer without causing any mutation in your primer. Okay, we already discussed a little bit here in the class before. Start and end with the 1 to 2 G and C pairs. Okay, this is more known as a GC clamp. Okay, the end of the primer should have around 1 or 2 G and C, okay, not more than that. If more than that, the binding of the primer to the DNA template is much strong, and then when the process of denaturation, it might not denatured properly. So this is important as well, the GC clamp. And then melting temperature, it should be around 50 to 60 degrees. And both pairs of primer should have within 5 degrees different, okay, between those primer in terms of TM. Okay, how to calculate the TM? Okay, you can see in the next link that I will provide you in these videos. And then make sure you go into the link to see how the primer design specifically, as well as tips how to design a good primer. Primer pairs should not have complementary region. Complementary region, that means if both primer, forward and reverse, have many complementary region in the sequence, it may bind to each other instead of bind to the template. So this can hinder the efficiency of the binding to the DNA template. So this should be avoided. Okay, we don't want to have a dimer primer because you will not have a good yield of PCR product for the after PCR reaction finish. Okay, here is some of the YouTube channel that you can go into, and how to do the location of the primer in the gene sequence, as also the primer design and tips. There are many applications why you want to use the PCR. Okay, PCR, medical application, okay, genetic testing to see whether the presence of the genetic disease mutation, tissue typing prior to the organ transplantation, formulation of individuals' cancer therapy treatments, forensic application, for example, the genetic fingerprinting. PCR can be used to incriminate or rule out the suspect in the crime investigation by comparing the bands of the molecular markers that you use in the genetic fingerprinting, whether you use the RAPD or RFLP and others. Okay, parental testing, PCR can be used to confirm the biological parents of the adopted child and or identify the remains of the unidentified body. Infectious disease detection and identification, in terms of the viruses, to detect, you can use the PCR. PCR is very sensitive and also very convenient ways to detect the viral loads in the sample, as well as in the very limited amount of the template available. And other disease organisms, such as those that cause middle ear infection, tuberculosis, or lyme disease. Early detection of several forms of cancer, including leukemia and lymphoma. Okay, these some of the biomarkers that available already been researched. Okay, there are several biomarkers that can differentiate the stages of the cancer progress in the patient. So with these biomarkers, you can have early detection before the cancer progresses. Detection of viral DNA and virulent subtypes, including those that cause the earlier epidemics. Okay, this happens in the COVID-19 as well. So you can see the how much the viral load in the patient through the real-time PCR, as well as antigen detection. Application in molecular biology research, DNA sequencing, DNA cloning, and gene expression, all these involve PCR in the process of gathering the DNA for analysis. PCR can be used to produce huge amount of pure DNA sample from a limited source. Production of hybridization probes for both northern and southern hybridization. Analysis of DNA from ancient source. Okay, one of the example using the PCR in diagnostic, okay, by using the real-time PCR. So the rapid result, okay, this one, whether you use the antigen base or the PCR, real-time PCR. Okay, real-time PCR, of course, it will cost more, it require longer time, as well as the, however, is much sensitive compared to the antibody or serotypes testing. Okay, primers can be used as a PCR process, or the probes for the detection. Okay, other than that, PCR or primers is used for the genetic engineering, okay, to make a modification on the certain organism. For example, here is microbes, okay, using the engineered bacteria for the fast, large-scale genetic manipulation based on the genetic parts that you can use, whether you want to overexpress it or suppress the expression of the proteins, so that you can have the full function of the bacteria and fully value-added products later after the research process. Engineering of microorganisms for the production of the biofuels, and perspective based on the system metabolic engineering approach. Okay, this is why we want to have genetic modification or genetic information so that we can manipulate it to get a better value of the products or the organism that you want to study. Okay, genotyping. Okay, genotyping techniques also been useful for the to see whether each the bacteria have developed a strain differences, and to detect the bacterium or in the effective treatments or to prevent the outbreaks. Okay, so the diagnosis of the bacteria also been used for the using the next generation sequencing or PCR chain reaction. Okay, this is some of the example of the applications of the PCR in the research been done worldwide. So because of you already finished the courses and all those things, so you might question yourself why you want to become a molecular biologist. Okay, so this is some of the research activities that involve in the, if you choose molecular biology as your field of interest. So you can do cloning and subcloning, synthetic RNA transcription, gene expression studies, okay, and then cell growth and development, see how the treatment of the drugs and others affected the organelle structure, cells assembly, and others. So DNA dynamics and chromosome structure, you might also want to involve in the omics technology, which is proteomics, metabolomics, transcriptomics, and whatever omics, genomics, and others. Okay, all these high throughput techniques you give can get many beneficial threat or things that you want to improve the industrial products. Recording and analyzing various type of data, and also application of research to solve health problem and cure diseases. This is more for the medicine or health related issues in the company related to the medicine and also pharmaceuticals. Okay, I think that's all from me. So I would like to wish you all the best in the next SAM, which is you're going to do the LI in the company that you choose. So make sure before you do anything, make sure you try hard to recall back what you have learned, and make use of all the skills that we have given to you throughout these courses, so that you can impress your employer and make a very good impression for UMP as well. Okay, and of course you want also to have a very good reputation um, for your product, for your work that you doing before maybe they can absorb you into their company after you finish the LI. Okay, with that, all the best, and see you later. Okay, bye.